Okay. Hi there, Epidemics. Hope you guys are all ready to talk about antibiotics and antibiotic resistance today. So I just want to remind you, there are no new assignments that are due this week. I will be posting a review quiz tomorrow, as well as your weekly journal assignment. And those will be due on April 14th. The project itself is due on Sunday, April 30th. If you're doing a presentation, you will do it May 2nd. Okay. So last time we met, we kind of went over some of the basics of bacteria, what they are. These little tiny, these are a type of bacteria right here. You can see this one is in the process of dividing into two. We talked a little bit about the two major categories, gram-positive, gram-negative, and basically what gram-positive and gram-negative means. So let me refresh your memory here. The difference between gram-positive and gram-negative has to do with the layers that are outside of the cell or plasma membrane. So all cells, our cells, plant cells, jellyfish cells, bacterial cells, have a plasma membrane or cell membrane that kind of holds everything inside, kind of creates that separation between the inside of the cell and the outside world. Bacteria have additional layers outside, including a cell wall for all of them, but the cell wall is different between gram-positive and gram-negative. And more importantly, gram-negative also have a second membrane on the outside. And that really impacts what kinds of material are able to get into the bacteria, and that influences which antibiotics are going to work best. Not to mention, some antibiotics work specifically on the cell wall or that outer membrane. And if you want to attack the cell wall, the gram-positive bacteria, their cell wall is exposed. It's easier to attack that. Okay, so antibiotics, we're talking, when we use the term antibiotics, anti is against, bio is life, but we're specifically talking about bacteria. Antibiotics are not going to work on viruses. They are not going to work on most parasites. They're not going to work on other kinds of diseases that people get, like heart disease or cancer. They're specific for bacteria. So this is a graph looking at the number of infections that caused death per 100,000 people in the US. So this is adjusted for the changes in population size. So this is kind of like a percentage of the population that died from bacterial infections. And as we can see, in the 1920s and 30s, here's our level up here. Then we developed an antibacterial group of drugs called sulfa drugs, and the rate of mortality with bacterial infections dropped dramatically. This was seen as a miracle drug. Then we developed penicillin, and after penicillin, the death rate plummeted even further. So when we look at this, a death rate that was in the high 200s plummets down here to about 50. Again, this is per 100,000 people in the American population. That's huge. That's dropping it by 200. And if you compare that to all other medical technologies, those other medical technologies, heart bypass surgeries, cancer treatments, over the next 45 years after we started having antibiotics, everything else only dropped deaths by 20 per 100,000. So that's a tenth of the impact that antibiotics had. We tend nowadays to take antibiotics for granted. And we tend to take for granted that most people are not going to die from infections. It's not that common. But before antibiotics, if we look back at World War I and earlier, more troops died from infection than any other cause in wars. That was the number one killer of troops, infections. And diseases were the leading cause of death for children. Over 25% of children died in the first year of life. 25%. This is what a world looks like without antibiotics. Nearly half of all children died before adulthood. Adulthood being usually considered 16 or 18, depending on the time period. But you're getting the idea. Huge numbers of children dying from infections because they don't have a fully developed immune system yet. Okay. When we talk about antibiotics, these are drugs that target bacterial cells by attacking something that those cells do differently than our cells. We're going to look at some of those targets, but I've already mentioned cell walls. Our cells don't have those. When we talk about antimicrobial or antibiotic agents, we can see what kind of effect it has on the bacteria. So we've got three different terms here. Bacteriostatic. So we've got bacteria and we've got static. Static in this case means staying still, unchanging. A bacteriostatic agent stops bacterial growth. So if we look at cells being grown in the lab, we can see that the cell population goes up, it gets treated with that bacteriostatic, the population stays the same, the bacteria are still alive. Bacteriostatic agents do not kill the bacteria, they just stop it from dividing. And if you remove that bacteriostatic agent, then the bacteria can start dividing again. So the population goes back up. What this is useful for is it keeps the bacteria at a steady level, and then it's depending on your immune system. Your immune system then comes in, it has the time to develop that more robust part of the immune response. Remember, The earliest immune response is fast, but it's not very powerful. And it's kind of like the second phase. It's very powerful. So it gives you time to develop that. And your immune system can then get rid of the bacteria. Then we look at bactericidal. Cidal, it's like homicidal. Cidal means death. So this means killing the bacteria. So a bactericidal, again, we see the bacteria going up. Then we see the bactericidal agent being delivered, and the number of live bacteria goes down. The bacteria cells are there, but they're dead, and they're not going to be able to divide or in any way. In order for something to be declared bactericidal in the United States, it must kill 99.9% of bacteria within 24 hours. So if you see something advertised as bactericidal, this is what they are supposed to accomplish in order to use that term on the label. So that should give you an idea, if you're looking at cleaning products, what this is. There's also one called bacteriolytic. This is sometimes used in cleaning, especially in places like hospital settings. Bacteriolytic, lytic means destroy or explode. And so that literally destroys the cell. So when you add a bacteriolytic agent, the number of cells goes down, dead and alive. Now, as usual, in biology, it's not always clean cut. When we talk about antibiotics, we can talk about antibiotics that maybe don't stop the growth, but slow it down to give your immune system time to catch up. Antibiotics that kill less than 99% of bacteria, but again, it's giving your immune system a chance to catch up. Okay, now yes. So when we look at antibiotics, there are 4 major targets. There are some other targets. You'll see I put a little blue box over this particular term. This is not a very common antibiotic on the market. So we're going to ignore that one. So the major 4 categories that we're going to talk about are cell wall. If you've got something that interferes with cell wall synthesis, synthesis is manufacturing. So that's going to stop cells from dividing. And penicillins and ampicillins and things like that target the cell wall. DNA synthesis, this is making a copy of the DNA. So again, in order to divide, bacteria have to make a copy of their DNA. If they aren't able to do that, they're not able to divide, and this antibiotic, again, keeps the population from increasing. So again, this is giving your immune system a chance to catch up. Metabolic enzymes. Okay, so that's not the best term for that. Well, I mean, it's a good science term. It's not the best term for you guys to understand. We're going to call it protein synthesis, making proteins. Proteins are a major component of our cells or bacterial cells. We manufacture the proteins that we need, and those proteins do most of the functions of the cell. So if you think about cells making energy, right? Or muscle cells contracting, or producing digestive enzymes. All of these things require proteins. So if you attack protein synthesis, that's attacking the way that cell functions. And so that's more likely to kill a cell. So the cell wall and the DNA targets, that's tending to stop cell division. So those are more bacteriostatics. This one will actually kill the bacteria. Oh, I'm sorry. Protein synthesis should be down here. So that's the tetracyclines. Up here is bacterial metabolism. So protein synthesis inhibition, stopping protein synthesis, stops all kinds of metabolic processes. These bacterial metabolism specific antibiotics target a specific process in that cell. So it may target energy production. It may target making carbohydrates or lipids. Just like proteins, we need to manufacture these. All cells need to manufacture these. It can also target the mechanisms that break down damaged molecules. So when we think about damaged molecules, we're talking about molecules that have gotten old, that have gotten hit by some solar radiation and damaged, things that their whole job is to act as a defense mechanism, but a lot of times that means that molecule, again, gets damaged by that process of defending the cell. If we don't break down those molecules, they build up inside of the cell and just kind of act like a traffic jam. Or more accurately, a hoarder's house. If you've ever seen reality television shows that talks about hoarders, you know that some of them just literally never throw anything away. And their houses get full of stuff until they have like little pathways. That's kind of what's going on if your cell cannot break down damaged molecules. It kind of gets filled up. And when you get filled up like that, every single thing you have to do is harder. Same thing with these bacterial metabolism targeting breakdown of damaged molecules. Okay. Questions on this?
Nope.
Okay. All right. So we're going to kind of walk through these major targets in a little bit more detail. So we're going to start by talking about cell walls. So what we have in this image, I will tell you that this website that I got this image from, they're not always accurate in their information. Don't think I'm encouraging you to use this learning app because it's a very bad one, at least for science. Okay, so here is the cell membrane, And here in a gram-positive bacteria, we have these peptidoglycans. Remember, peptide means small protein, glycan means carbohydrate, and these are all linked together. So all of these different layers are attached to each other. Certain types of medications or antibiotics, like penicillin, will block cell wall manufacturing or repair. And different antibiotics may target different steps, but for example, they may block making peptidoglycans, or they may block the cross-linking process. So if you are infected with a gram-positive antibiotic, this works great. So these are things like staphylococcus infections, or I guess that's the example I have. These have low side effects because they're tackling something that we don't have. We do not make peptidoglycans the way bacteria do. These do not work on gram-negative bacteria, and they do not work on, did I put a list of these antibiotics? I don't think I've made a list of which antibiotics these are, but these antibiotics that work against gram-positive bacteria will not work against gram-negative, and they will not work against things like tuberculosis, which I'm just going to abbreviate TB, which is considered a microbacterium. It's not gram-positive or gram-negative. It's in a different category altogether. So, oh, here we go. Penicillin, cephalosporin. Those are your examples for gram-positive specific antibiotics. Oof, this looks confusing, doesn't it? If we're looking at things that target protein synthesis, so I'm not expecting you to look at this and understand exactly what's going on. So let me walk you kind of through this. The process of making proteins, you may have heard about this. DNA is copied to what's called mRNA, which is messenger RNA. The mRNA then goes to the site where there are ribosomes. Ribosomes are complex molecules. They've got proteins, they've got other kinds of molecules in there, but that is where protein manufacturing occurs. So in simplified terms, your messenger RNA is in between these two parts of the ribosome. These parts of the ribosome can read the information on the messenger RNA and convert that to an amino acid sequence. And proteins are made of strings of amino acids. So basically, you're converting that information on the mRNA into how to make that protein. And in order to do that, there are pockets on the ribosome where molecules can bring in the new amino acids to attach to that growing peptide chain. So they're bringing in the new material to add to that chain. And things like tetracycline can fit into that pocket, which means that this new amino acid cannot be brought in. So tetracycline literally clogs up the machinery that manufactures proteins. Now, if you're blocking manufacturing, you can block RNA manufacturing, and that will stop the stop protein synthesis or manufacturing. So the bacteria cannot make proteins. You can block the ribosome or attack the ribosome, and again, you're stopping protein manufacturing. Okay, so our example antibiotics are things like streptomycin, tetracycline, doxycycline, there we go. Let's get the spelling correct. Now, these are wide spectrum antibiotics. So it doesn't matter so much if it's gram positive or gram negative. The downside, the big downside to these is they damage the microbiome. So have you heard this term before, microbiome? Does anybody know what that is?
Isn't it just all of the bacteria that lives within our body?
Yes, exactly. It's all of the bacteria that live on and in our bodies. And we need the microbiome to survive. If we got rid of the microbiome, we would not be able to survive long term. We would have lots and lots of health problems. We'd need lots of supplements. It would not be a good thing. And so if you've ever taken streptomycin, tetracycline, doxycycline, and then ended up with stomach problems, that's from the damage to the microbiome. I mentioned DNA replication. That's a fancy word for copying the DNA. So there are a few, so bacteria do this in a different way than our cells. And they use different enzymes. Enzymes are just proteins that do things in the body. So enzymes that are involved in DNA replication will be involved in matching up the newly growing strand of DNA with the existing strand of DNA, making sure that they are identical, making sure that the right new bits of DNA attach to the old bits of DNA. I'm trying to make this very simple. Now, there are not many bacteria that target this, or antibiotics, sorry. They are generally bacteriostatic. So they don't kill the bacteria, but they stop DNA replication. Just like with the cell wall, when you block manufacturing of the cell wall, that's primarily a bacteriostatic event. If you're blocking protein manufacturing, That's bactericidal. That's actually killing the bacteria. And the last category that we're going to talk about is bacterial metabolism. Again, there are not a lot of antibiotics in this category. One of the targets is folate synthesis. So folate, also known as vitamin B9, this is something that we need in our diet. And one of the places we get it from is our microbiome. We actually digest some of those bacteria and take up that vitamin B9. It's important for our regular life, but it's also very, very important during pregnancy. So pregnant women often get folic acid as a supplement to help with the developing embryo. They need more than the average person does. A vitamin is a small molecule that we cannot make ourselves. We have to get it from our diet or our microbiome, or a combination of the two, really. So if we block folate synthesis in bacteria, the bacteria are not able to copy its DNA. So folate is important in DNA copying. So if that is our target, it kind of does the same thing that blocking the enzymes involved in DNA synthesis. It really just stops dividing. So again, it is bacteriostatic. So kind of summarizing this, the most common antibiotics are going to go against cell walls or against protein synthesis. And the other categories, not quite as much. Okay. So how do we decide the antibiotic? Well, we have to ask a couple of questions. What type of bacteria? That's the big one, right? Because if you're going to target the cell wall, it kind of needs to have the exposed cell wall. It needs to be in that particular category. If you've got tuberculosis, you're going to need a different antibiotic because it doesn't have that same exposed cell wall. You can kind of get a guess. Well, if you've been to medical school, you can kind of get an educated guess based on the symptoms. Where is that infection? What kind of symptoms is it causing? Where did it come from? So this is a great question. Did you catch this bacteria in Boston at a probably at a hockey game or a basketball game? Or did you just come back from a trip to Egypt? If you do international traveling, always tell your doctors. Very important. And how severe are the symptoms? So severe sickness, people who are really, really sick, they generally start with broad spectrum antibiotics. they do this because if the symptoms are severe enough, you've got to do something to keep the person alive until you can figure out what bacteria it is. So they'll try and culture the bacteria, which is a fancy way of saying get it to grow in the lab and identify it. Gram staining is one of the earliest processes to identify at least big categories of bacteria. There are much more advanced techniques now, including analyzing the genetic material, the DNA of the bacteria, seeing what kind of bacteria it is. Once they identify the bacteria, they'll often change what antibiotic they're using, trying to get the most specific antibiotic for that bacterium and save those broad spectrums for when they are absolutely needed. So we don't want to start everybody out on things like vancomycin. We want to hold some of those antibiotics back. If we can make do with something easier. So, where did these antibiotics originally come from? You probably heard this story. Alexander, I think he spells his name, I'm not sure. Fleming discovered penicillin in 1928. This is a well-known story. You may have heard this about how Fleming left a petri dish. This is a petri dish with bacteria on his lab bench. Some mold spores blew in the open window and landed on the petri dish. He came back from vacation and noticed the bacteria around the mold had died. Okay, so this is one of those stories that in science we call these just so stories. It's a nice, pretty little package with nothing complicated about it. And that's often a good sign that it's not completely accurate. So this is Fleming trying to look intelligent. He was intelligent, don't get me wrong. This is a petri dish. It is a semi-solid material. It's a plastic dish or a glass dish filled with a semi-solid material that has food for bacteria growth. And you streak the bacteria across the plate. So you use a special tool and you can really see the streaks, right? Spreading back and forth, back and forth, back and forth, back and forth. This one was very, very heavily seeded. They put a lot of bacteria on this plate. Here is your penicillin mold. And you can see there's an area around that mold where there's no bacteria growth. So that's what this looks like on a petri dish. So why do we say Alexander Fleming discovered penicillin? And I use my scare quotes. And why do I call it a just so story? Long time before Fleming did his experiments, many different cultures would use moldy bread to treat various ailments. Often they would do these topical on infected wounds. Sometimes they would eat the moldy bread, especially if they had things like urinary tract problems or digestive problems. And when I say many cultures, we know that this happened in Egypt, China, Serbia, Greece, Rome, and pre-Columbian Central America. So it's not just a couple of places. It was a widespread tactic. What Fleming did with his work was allow us to extract concentrated penicillin, the active molecule that caused the inhibition of growth. So Fleming did this in 1928, but from the 1800s, there are publications that show bacteria will not grow on moldy petri dishes. And in fact, the bacteria that we get penicillin from had already been identified as a specific bacterial inhibitor. So Fleming gets the credit for his discovery, and certainly being able to extract the penicillin was a huge advantage, but it's, standing on the shoulders of giants. This is always how science works. We tend to take one individual and put them up on a little pedestal and say, look what this person did. And it's always more complicated than that. So before penicillin was developed as a drug, in the 20s and 30s, the 1920s and 1930s, a group of researchers found that a synthetic dye that contained sulfur could treat bacterial diseases. How they figured this out, I haven't found that story, but that's a pretty impressive finding to make. You're making a real leap from making something to dye clothing with and treating patients. These sulfa drugs, named because they have sulfur, reduced death to what we called childbirth infections, which is a specific type of infection, from 25% to less than 5%. So when we looked at that graph and we saw sulfa drugs and a huge drop off in mortality rate, this is what they were treating, a specific infection that was called a childbirth infections, or a specific group of infections. Now, on the downside, this was something that had been discovered a long time before. It was officially off patent, so they couldn't patent it for this use. Why does this matter? It means that the drug is in the public domain, and anyone can manufacture it. This particular one was also very easy to make. So you had lots of people manufacturing this and selling this. In addition to this being off patent, so anybody can make it, it was also basically unregulated. We have a lot of things that are for sale in our grocery stores and our drugstores that are basically unregulated. So I think we've talked about vitamins, herbs, Those are basically unregulated in the US. There is no rule that says that it has to be what it says or that it has to do what they claim it does. The only restrictions on it is that it should not cause harm. However, there's not a lot of monitoring for that either, especially long-term effects. So at this time, all drugs were like that. Nothing was regulated. And during the 1930s, a company made a sweet tasting syrup of this sulfa drug. And to make it sweet tasting, they added something called diethylene glycol. Diethylene glycol is an antifreeze. So if you've ever, antifreeze is something that's used in vehicles specifically to prevent water that's used for cooling the engine from freezing. Antifreeze is deadly, and it's very attractive because of the taste. So it's commonly cleaned up. If there's a leak, animals often get into it and they will lick it up, and then a lot of them don't survive, or if they do survive, they have lots of very expensive vet bills. So here we go. Here's a company putting this poison in a medication. Fortunately, the newly developed FDA was able to stop manufacturing and collect almost all of this medication. The company was sued. The only reason that they were able to be sued was for mislabeling. Because the label called this an elixir, and the law required elixirs to have alcohol. So that's it. That is the whole reason they were able to be sued. Not because they put a poison out, not because it was potentially deadly, but because they called it an elixir, and the law required elixirs to contain alcohol, and it did not contain alcohol. If they had labeled it a syrup, there would have been no legal recourse, even if it had killed people.
That's crazy.
Yes, it's absolutely crazy. As a result of this event in 1938, the government passed the Food, Drug, and Cosmetic Act, which added regulations to food, medications, and cosmetics, and started adding oversight into how medications were manufactured. So it really gave some power to the FDA to regulate medications. So this is a good story because, as far as I can tell, nobody died from this incident, but it could have been very, very tragic. Unfortunately, resistance to these sulfa drugs showed up relatively quickly, but we have penicillin coming down the line shortly after these sulfa drugs, the resistance to these sulfa drugs starts showing up. Oh, here's a bottle of that elixir that had the diethylene glycol in it. All right. So after Fleming said, okay, here's this molecule, penicillin, that can stop bacterial infections. But it took a long time for chemists and microbiologists to develop a method to grow the mold and extract and purify the penicillin. So remember, Fleming did his announcement about penicillin in 1928. The first person to receive penicillin, Ann Miller, received it in the 1940s. Actually, sorry, the first person to receive penicillin was not Ann Miller. Ann Miller was the first person. We'll get to Ann Miller's story. So the first person who received penicillin was a police officer. And this police officer had a rose garden. So in his spare time, he grew and, grew roses. Roses have thorns, right? Well, he got a scratch. Excuse me. And while these days we don't think anything of that. I'm a gardener. I've always got scratches on my hands. But at this time period, you could get infected. And if you got infected from just a scratch from a rose thorn, you can have a horrible reaction. And this police officer developed pustules, kind of like buboes or ulcers, open, oozing, pus-filled pockets all over the body, a raging fever. He had clearly gotten a systemic or body-wide infection. He was dying from a scratch from a rose. So they had enough penicillin, or they had some penicillin, and they're like, okay, we're going to try and treat him with this. So they gave him penicillin. It reduced those pustules. It brought down his fever, but they ran out of penicillin. And he ended up dying from the infection. So they just didn't have enough of it, even despite the fact that they'd been working on it for over a decade. And they'd been stockpiling their supplies for over a decade, and they didn't have enough to treat him. In 1947, Ann Miller had a miscarriage. And as a result of this miscarriage, she developed an infection in the blood, or what was at the time called blood poisoning. She was running very, very high fevers. Her doctor got hold of 5.5 grams of penicillin, which was half the world's supply. The doctor guessed at the dosage. No one had any clue how much penicillin a person needed to take. So they gave Miller some penicillin. She survived the night. This was great, right? And then she survived for the next 57 years. She was the first person whose life was saved by penicillin. We knew her name. How crazy is that? We often don't know the first person who was successful at something like surviving a massive infection, but we do know her name. So that is the start of penicillin and the start of the antibiotic era. Any questions on that part? Okay. So I've already mentioned this one, the microbiome. And you guys already know that it's really all organisms living on or inside the body. So it's bacteria, it's fungi, it's viruses. We even have little teensy tiny mites, which are kind of an insect that live on our faces. They especially right around the eyes. They like to live inside the hair follicles of your eyebrows and your eyelashes. And of course, I can just picture people shuddering because I've seen students shudder at this point. Those little mites are actually eating up dead skin cells. They're eating up oils that you produce. And this actually helps prevent bacteria from being able to use those food sources. So you kind of want your mites. We think there are trillions of individual microorganisms, not trillions of mites. Don't worry about that. We think there are thousands of different species. It's really recently started to be discovered. So we don't have actual numbers. We do have researchers who are looking at different microbiomes in different people, but they are so complicated and there are so many differences between people that it's going to take a lot of work and a lot of time to figure out this species is good for you because it helps protect you from this disease, or this species is not so good, it makes you more likely to get that disease. So we don't have a clue about any of that part yet. Each person has a unique microbiome. So it's a unique collection of organisms. You are going to have certain organisms that your parents do not have, that your partner does not have. Our life experiences impact what we have as our microbiome. We get our first microbes during birth from mom. And we do know that vaginal birth does a better job of getting our new microbiome started. Children who are born with C-sections tend to have less diverse microbiomes. And we think that some of the minor, I'm saying minor health issues that are more common in C-section babies, things like allergies, we think that that is because they didn't get as good of a exposure to the mother's microbiome during the birth process. But we also pick up microbes from other people, from our pets, from the environment. And we know that people who are exposed to pets, the outdoors, during childhood, have things like fewer allergies as adults. So we know that these have a protective effect, and that having more exposure to harmless microbes helps protect you later in life. And of course, we lose some microbes. We might lose some to infections. So a different bacteria comes along and kills off one species in our digestive tract, for example. Different medications, antibiotics, tells you right there on the label what it's doing. It's impacting your microbiome. Chemotherapy will impact your microbiome. There are some other medications that can be involved in this as well. We know that the microbiome is critical for our survival. I mentioned that earlier. So let's talk about some more specifics. It's involved in regulation of the immune system. So it does help defend us against foreign microbes right off the bat, but it also helps our immune system learn how strongly to respond to a new invader and how to recognize a new invader versus something that's basically harmless. So it's training our immune system. I mentioned that we get vitamins from them. We get a bunch of the B vitamins, we get vitamin K. So these are things that we have to have, that we cannot produce ourselves, and we get from our microbiome. They also provide some amino acids and some what are called fatty acids, which make up lipids or fats. Again, these are things that we can't manufacture. And these bacteria, this microbiome in our gut, either manufactures these directly or modifies our food to allow us to take them up and absorb them. All right. So when we talk about antibiotics, we need to be aware of the impact on the microbiome because the microbiome is so beneficial for us. If you have seen products that are supposed to support your microbiome or probiotics, which are supposed to provide you with bacteria, I want you to remember this part right here. Oh, that was a lousy jump drawing. Try this again. Let's just draw a line under it. Your microbiome has thousands of different species. Probiotics generally have a few. I think I've heard some bragging about having maybe 100. That's not to say there's not a role for probiotics, but it's just a drop in the bucket compared to what we already have. What we know about probiotics as far as health is that they are useful after taking antibiotics or after treating a fungal infection. Both of these conditions, fungal infections being yeast infections, jock itch, athlete's foot, probiotics will help your microbiome recover after these events that are going to impact your microbiome. In fact, a lot of people who are vulnerable to fungal infections will find that they take antibiotics and then they get a fungal infection. So probiotics can help prevent that. So there is a role for them, but you don't need to take them on a regular basis. Questions on probiotics? Whew. here we go. Another complicated graphic. So you can see this black arrow that runs from 1900 to 2010. And what we see is kind of the history of antibiotics. So we really started looking at antibiotics or antimicrobials in the early 1900s. And we actually did develop an antibiotic in the 1910s that was not very successful. Penicillin was really the first successful antibiotic. Where's my arrow? And that was right here at 1928. Remember, it wasn't used until 1947. So what we have is we have this time period where we're slowly making progress, slowly developing and discovering new medications, starting to see resistance showing up pretty quickly. And then in the 1940s, we really start getting a lot of different antibiotics being discovered and started getting them used. And from the time penicillin was first approved for use in people, for the next 20 years or so is what we call the golden age of antibiotics. So here's a whole bunch of antibiotics that were developed. Here's another whole bunch. Here's a bunch in the 40s. They didn't get approved until a little bit later, but people were starting to work on them. In the 1960s, we first spotted MRSA. MRSA is a streptomycin. Wait a minute. A strep bacteria, Streptococcus, there we go. Aureus. Aureus means yellow. It's yellow when you grow it in tissue culture. And I'm sure that that is not spelled correctly. It's not strep either, it's staph. Oh, man. Blah. Must be getting to the end of the lecture. Staphylococcus aureus. And MRSA is resistant to many different antibiotics. And it's a nasty infection when you get it. So we all have staph bacteria on our bodies all the time. But when we get a staph infection, we can get skin ulcers. We can get things that look a lot like buboes. And it's hard to treat because it is so resistant to antibiotics. And this was discovered, it was seen for the first time in the 1960s. So between the 1960s and about the year 2000, we didn't have, we didn't have a lot of new antibiotics, especially not new types of antibiotics being developed. We're now trying to go after different targets. So no longer just going after the cell membrane, easy target, or protein synthesis, pretty easy target, relatively easy target. We're trying to go after metabolism, or we're trying to go after DNA replication, or maybe something else. The tricky thing is the first drugs developed are often the low-hanging fruit. So the first antibiotics went after the things that were easiest to go after. We picked all the fruit that hangs low off the tree, and now we've got to climb the tree to get more fruit. And it's going to be complicated, and it's going to be more challenging to go on from here. Most of the antibiotics developed since the 1960s have just been the same targets. They're just different versions of the existing antibiotics. So if we are no longer able to go after the low-hanging fruit, we have to have new discoveries. This isn't very well funded. This would have to be funded by the government because it's very expensive to do this. And drug companies are not likely to go after that. Antibiotics are cheap and taken short term. So there's not a big return on investment. Drug companies are not looking to go into these fields because they're not going to make a lot of profit off of it. So, you know, that is kind of where we are problematically. We're not getting new ones developed, and we're getting a resistance to existing antibiotics. And that's what we're going to talk about next week, is the antibiotic resistance problem, why we have an antibiotic resistance problem, and hope for the future, where we might be able to go next. Any questions on anything we talked about today?
Awesome.
All right, if there's nothing else you guys want to know today, have a good weekend, and I'll see you guys on Tuesday.
Have a good
Thank you.
weekend.
You too.
Bye.
Bye.